this video, let us go some detail into the receptors that we have studied in the previous video. The first thing is ligand-gated ion channels. Here in ligand-gated ion channels, let us consider a structure. This is a receptor that is present on the cell membrane, and this is a place where a ligand can bind. This is the ligand binding site of the receptor. So as soon as we take the drug, the ligand binds to the ligand active site, binding site, and this ligand binding allows the ions to pass through the receptor. Sorry for the disturbance. After the ligand binds to the receptor, it allows the ions to pass through the receptor. It allows those ions which are structure specific ions only. Then after the ion enters into the membrane, it causes depolarization. So here what we have written is ligand is hydrophilic. As the ligand is hydrophilic in nature, so it cannot pass through the membrane, so it binds to the receptors that are present on the membrane. Thus it cannot cross membrane. So what are the action that it brings to the membrane is it changes in membrane potential due to ion exchange. So changes in potential due to ion exchange, either repolarization or depolarization. Next thing is G protein couple receptors. These are the G protein couple receptors. It consists of 7 alpha helix cylindrical structures like 1, 2, 3, 4, 5, 6, 7, and these are bounded by extra cellular loops and intracellular loops, and this also contains alpha, beta, and gamma proteins. So it has a ligand binding active site here, and after the ligand binds to the G protein, then the alpha protein in the G protein couple receptor, it what it does is it transforms GDP into GDP by phosphorylation. This GTP causes the biological response, further biological response. So ligand binds, then GDP is converted to GTP. Ligand is hydrophilic, so cannot cross the membrane, so it binds to the G protein couple receptors which is present on the membrane. So what are the action that it brings is phosphorylation of G protein. Next is enzyme linked receptors. It is an enzyme and it is the binding site. So after the ligand drug enters into the body, it binds to the enzyme binding site. Then what action it does is phosphorylation of proteins and receptors. So what it does is, the example is ADP to ATP. It performs the action like kinases, that is, it adds phosphoryl group to the R group, that is R is converted to RPO4. That's R is got phosphorylated. So ligand binds, enzyme activates. So after the ligand binds to the enzyme, then this enzyme gets activated and this enzyme does its respective functions. So what are the action that it brings is phosphorylation of proteins and receptors. And the last thing is intracellular receptors. Intracellular receptors, those are present inside the cell. So this is a cell membrane. After the ligand approaches the membrane, as it is hydrophobic in nature, it crosses through the membrane and it binds to the intracellular receptor, and this drug and intracellular receptor combinedly enters into the nucleus and they bring gene expression, alteration in gene expression, like this we can consider in anti-neoplastic agents like alkylating agents. They bind to the nucleotides of the DNA. It's some kind of example. So here ligand is hydrophobic, protein phosphorylation and gene expression altered. These are the classification of receptors I have presented in this way, so that people who are aspiring to give good presentation in the exam, it will be very helpful and it aids the student to secure good marks. Thank you.